Hello, everyone. I would like to start by thanking the organizers for inviting me to this conference and giving me this opportunity to present our recent work. My name is Yunhee Jeong, and I'm a senior scientist in immunology and immunosafety group at AbbVie. Today, I'm excited to share insights that our group discovered during the development of in vitro immunogenicity assays. Our team focuses on assessing in vitro immunosafety in the preclinical stage, and I will discuss how we established endotoxin limits to enhance the reliability of our immunogenicity risk assessment. Here's what I will cover today. I will talk about what immunogenicity is and why in vitro assays matter. Then I will explain the assay and how it links to clinical outcomes. We will see how endotoxin levels affect accuracy and responses. Lastly, we will discuss why setting endotoxin limits is important for developing biotherapeutics. I aim to show how these assays improve drug development. Immunogenicity is the propensity of the therapeutic product to generate immune responses to itself and to related proteins, or to induce immunologically related adverse clinical events. It's a big concern in drug development because it can affect how a drug works and its safety. Regulatory agencies like the FDA and EMA provide guidelines for immunogenicity assessment to ensure the safety and efficacy of drugs. These guidelines specifically require the measurement of anti-drug antibodies as part of the process. Understanding and effectively measuring immunogenicity is essential for improving patient safety and drug performance. Immunogenicity can result from a range of factors related to both patients and products. Patient-related influences include genetics, immune status, and specifics like dose and route of administration. On the product side, factors involve structural properties, glycosylation, impurities, aggregates, and even unknown factors. These varied factors make predicting immunogenic responses quite challenging. However, understanding this complexity is crucial for developing effective strategies. By acknowledging these influences, we can strategically mitigate immunogenic risks, ensuring the development of safe and effective therapies. Strategic planning is key to navigating these complexities in drug development. This slide highlights the importance of T-cell responses in immunogenicity, particularly involving antigen-presenting cells. When a biotherapeutic is administered, antigen-presenting cells process and present the therapeutic as epitopes to T cells. Depending on the patient's MHC class II type and the specificity of these epitopes, CD4 T cells can become activated. This activation is a crucial step in triggering an immunogenic response. Our in vitro assay measure the activation and proliferation of CD4 T cells, enabling us to predict immunogenicity responses. By assessing the extent of T-cell activation and proliferation, we gain valuable insights into the potential immunogenic risk. In vitro immunogenicity assessments are crucial tools in drug development. They offer a controlled environment to study the immune response potential of therapeutic proteins early in the drug development process. By enabling predictions of immunogenicity through CD4 T cell proliferation assays, we benefit in several ways. Most importantly, these assays allow us to identify and address immunogenic risk before clinical trials, improving both the safety and efficacy profiles of biotherapeutics. Moreover, using in vitro assays for prediction can lead to significant cost and time savings in drug development. Ultimately, the ability to predict immunogenicity responses with in vitro assays enhances the overall drug development strategy, leading to safer and more effective therapies for patients. Our developed assay is a flow-based method focusing on CD4 T cell proliferation. This approach allows us to accurately measure the degree of T cell activation in response to a given biotherapeutic. To quantify this response, we calculate the stimulation index, which is the ratio of proliferated T cells to those in the media-only condition. This ratio helps us determine the extent of immune activation. We define donors as responding donor if their stimulation index is 2 or higher. In this slide, we present the results of testing several biotherapeutics with known clinical ADA benchmarks using our assay. This assay allows us to analyze individual donor responses, drug-specific responding rates, and combine these into an overall response index. By comparing these metrics, we gain insights into how each donor and drug performs, which help us understand the immunogenic potential of these therapeutics in a more detailed way. Building on our findings from the previous slide, we observed that the results from our in vitro assay showed a strong correlation with known clinical ADA rates. This correlation emphasizes the predictive power of our assay. Now I will show you how T-cell proliferation responds in a dose-dependent manner to varying levels of endotoxin. Our research demonstrates a compelling pattern. As endotoxin concentration rises, we observe a corresponding increase in T-cell proliferation. At a concentration of 3 EU per mg, all donors exhibited immune responses, highlighting the potent immunogenic nature of endotoxin. This dose-dependent response underscores how vital it is to recognize the influence of endotoxin contamination on these assays. It underscores the need to tightly control endotoxin levels so that in vitro immunogenicity assessments accurately reflect a drug's true potential without any interference from contaminants. I'd like to highlight how endotoxins lead to the expansion of myeloid cells. This expansion occurs in a dose-dependent manner. As the level of endotoxin rises, so does the activity of myeloid cells. This is primarily due to the stimulation via the TLR4 signaling pathways known for triggering strong immune responses. On the first day, we observed the activation of inflammatory pathways by secreting these innate inflammatory cytokines. By the seventh day, this resulted in the release of T-cell-driven cytokine like interferon gamma. These observations emphasize the importance of precisely controlling endotoxin levels to prevent them from skewing the results of in vitro immunogenicity tests. Here, I am presenting our findings from comparing antibodies with different endotoxin levels. The study involved testing various lots of the same monoclonal antibody, each with different level of endotoxin concentrations. Notably, Lot 3 had been remediated for endotoxins from Lot 2. This comparison clearly showed that higher endotoxin levels led to increased T-cell proliferations. These results highlight how even small variations in endotoxins levels can significantly impact immunogenicity assessments. This shows that controlling endotoxin levels can greatly influence the accuracy of immunogenicity measurements. I'd like to summarize the key points of my presentation. Our research indicates that residual endotoxin levels can greatly impact the accuracy of in vitro immunogenicity assays, especially T-cell proliferation assays, by potentially causing false positive results. By effectively controlling endotoxin levels, our in-house assay can accurately predict immunogenicity risk and shows strong alignment with non-clinical ADA outcomes. We recommend setting a standardized endotoxin limit below 0.1 EU per mg in these assays to improve data accuracy and reduce false positives. Additionally, further research with a broader range of biotherapeutic drugs and a larger pool of donors is necessary to validate our findings and adapt them to various therapeutic settings. Establishing endotoxin limits enhances the predictability and reliability of in vitro immunogenicity risk assessments, improving the prediction of clinical outcomes. This study also contributes to regulatory guidance by proposing specific endotoxin thresholds for immunogenicity assays. By conducting more accurate assessments, we can enhance drug safety and efficacy and minimize the risk of ADA-related adverse effects, ultimately saving time and resources in the drug development process. These findings provide a foundation for standardizing immunogenicity testing protocols across the biotherapeutic industries. Lastly, I would like to take a moment to acknowledge the incredible team and collaborators who made this research possible. This research was primarily conducted by the immunology and immunosafety team, whose dedication and expertise were fundamental to its success. The in vitro immunogenicity working group provided immense support throughout this project. Lastly, I extend my gratitude to our leadership in the LDTS and biologics development science for their guidance and support. As we wrap up this presentation, I welcome any questions or comments you might have regarding our research. Please feel free to reach out via email if you'd like to discuss this further. Thank you for your attention and interest in our work.